Welcome to the third video in the unit on bacterial genetics. In this video, we're going to look at how we go from DNA to a functional gene product, which is typically the proteins. And so throughout this video, we're going to talk about the process of transcription and translation. I'm going to expect you guys to be able to use a codon chart and do translation, but for this class, I'm not going to expect you guys to be able to do transcription. I'll provide you messenger RNA sequence and just ask you to be able to translate that messenger RNA into a protein. And we'll go over how to do that in this video. So let's go ahead and get started. One of the important functions of DNA is to store the information necessary for the cell to function. The transfer of this stored information from the DNA to the functional gene products is called gene expression. Those functional gene products are typically proteins, but not always. There are some RNAs that are the final product that are going to do something in the cell. These functional products are what give cells their traits and their phenotype and really allow them to function. So gene expression is going to begin at a promoter sequence. It's this promoter sequence that signals for the start of transcription to occur to make the RNA itself. It's important to note that only one strand of the DNA is actually transcribed. It's called the template strand, and this is what is read to form the complementary base pairs that go into the RNA sequence. The other sequence of the DNA strand is called the sense or the coding strand, because this strand is actually going to match the RNA code. After we do transcription, if this is messenger RNA, then gene expression continues on to the translation of a protein. We're going to look at the details of all of these steps over the next couple slides. You should already be familiar with the process of transcription as the copying of DNA information into a sequence of RNA. Remember, we typically think of transcription as making messenger RNA, but all types of RNA found in the cell must be transcribed from a gene. Transcription in bacterial cells requires two proteins that interact together in a structure called the RNA polymerase holoenzyme. The first protein of the holoenzyme is the sigma factor. This is a DNA binding protein that recognizes the specific spelling found in DNA in two locations upstream or in front of a gene. The two locations are the key part of the promoter sequence, that sequence that indicates the presence of a gene and says start transcription here. These sequences are found 10 base pairs and 35 base pairs upstream of the start of the gene, and so they're called the -10 and the -35 sequences. The second protein of a whole enzyme is the RNA polymerase itself. This is the DNA-dependent RNA polymerase that will use the DNA as a template to build a new strand of RNA. After binding to the promoter, RNA polymerase is able to separate the two strands of DNA, similar to helicase in replication, and then reads the template strand to build RNA as it travels along towards the terminator sequence. The sigma factor is going to detach from the RNA polymerase because it's just a binding protein and not an enzyme. We'll add some sequence to this DNA molecule to remind you how transcription works. So here's our double-stranded DNA molecule with the complementary base pairs. As RNA polymerase was traveling along, it's going to be reading this template strand, which is the strand that's going to go 3' to 5'. But the coding strand is the one that goes 5' to 3' because it's going to match the RNA code, which also goes 5' to 3'. And so as we make the RNA sequence, if there's a T on the template strand, RNA polymerase puts in the complementary base pair into the RNA, which is an A. If there's an A, the complementary base pair, remember, for RNA is U for uracil because there's no thymine in RNA. A G will bring a C will bring a G, and a G will complementary to a C, so on and so forth, until we get to the terminator region where RNA polymerase will leave and will stop our RNA sequence. If you notice, the sequence we have generated is the same sequence as the coding strand, except the T has been replaced by U's. This is why it's called the coding strand, because it does match the code of that RNA transcript. I mentioned previously that it's not just messenger RNA that gets transcribed. There are also genes in the genome for other types of RNA, and these genes have their own promoter regions to initiate transcription. However, they don't have a ribosome binding region because the RNA is the final product. They're not actually translated into protein. So messenger RNA is the only type of RNA that contains a ribosome binding site because it's the regions of RNA that come from coded genes that are going to be translated into a protein. Transfer RNAs function in translation, but they're responsible for bringing the amino acids to the ribosome so that those amino acids can be put together and built into a protein chain. The ribosomal RNA is actually physically part of the ribosome itself, and it's actually the ribosomal RNA that's going to take those individual amino acids and attach them together with covalent bonds to make the growing peptide. This is a little bit different because most of the time proteins are the enzymes that do these types of chemical reactions, but the ribosome is different. The proteins of the ribosome are just there for structural components. It's the ribosomal RNA that's actually catalytic, that's catalyzing the connection of the amino acids together. RNA that's like this, that can function like an enzyme, are called ribozymes. And then the last type of RNA are called microRNAs. These are small little RNA sequences that we're just really learning about now that seem to be involved in the regulation of this gene expression, so controlling when and how much gene products are actually produced. Once messenger RNA has been transcribed, it will be used to build protein sequences through translation. However, there are 4 different nucleotides and 20 different amino acids. So the nucleotides need to be read in groups in order to have enough combinations to encode for all the amino acids. The correct grouping of nucleotides is every 3 nucleotides is going to equal 1 amino acid. The groups of 3 are referred to as codons. Codons are organized in a sequence one after another and do not overlap, so they must be lined up properly to form the correct sequence of amino acids. The lining up of the codons is called the reading frame and is set by what we call the start codon. This is the codon that indicates the protein should begin there and sets the proper grouping of 3 for the rest of the protein's reading frame. Translation then continues until you reach a stop codon, which is a codon that does not code for an amino acid, and so it indicates the end of the protein sequence. You should understand how to translate a sequence of messenger RNA into a protein using what we call a codon chart. This codon chart indicates what amino acid is encoded by each codon. So for example, if you saw UUU as a codon in the messenger RNA, that would indicate that phenylalanine amino acid should be added to that spot in the protein itself. The only codon you need to memorize is the start codon, so you know where the protein begins. The start codon is always AUG, which encodes for methionine. So let's use this DNA sequence to walk through the process of translation. The DNA sequence has the end of the promoter here in bold, so the -10 and -35 sequences would be somewhere up here in the sequence. And then it's got the transcription terminator here in red. And we can see the transcription started right after the promoter, and where we had a T in the template strand, it got encoded into an A, so on and so forth, till we got to the terminator sequence. Now we are going to use this messenger RNA and the codon chart to determine what protein would be encoded by this piece of nucleotide. Remember, the first step is to figure out your reading frame, because if we're going to read in groups of 3, this could be a reading frame, or that group of 3, or that group of 3. And depending on which group of 3 you were using, it would change how you read the entire sequence. This is why we need to remember that AUG start codon. So we're going to scan through our messenger RNA sequence until we find the AUG. Once we have that, now we can set up our group of 3 codons that are going to indicate the reading frame of this protein. And for each group of 3, we find them in the codon chart. So AUG, we said encoded for methionine, so that would be the first amino acid in the protein. CUU, if we look up into the chart, CUU encodes for leucine. ACU, if we look into the chart here, encodes for threonine. GAU, here is asparagine. And then UGA, when you look at the chart, you'll notice says stop. You can either write stop here, or you just stop making it, the protein, and now you have a protein that is made up of 4 amino acids that are connected in this way. Well, you have a nice easy codon chart to read to determine what codon equals what amino acid. The cell does not. Instead, its codon chart is the transfer RNAs. These are how the cell connects a specific codon in the messenger RNA to its corresponding amino acid. It does this because it contains what's called an anticodon region here at the bottom of the molecule. That anticodon, you will notice, forms complementary base pairs with the codon itself. So where the codon reads GCC, the anticodon is CGG to form the complementary base pairs. The tRNA with this specific anticodon will be carrying the amino acid that corresponds to the codon itself. For example, if you look back at the codon chart for the GCC codon, you will find that it codes for the amino acid alanine. This means that this specific tRNA with the CGG anticodon will always have an alanine amino acid attached to its amino acid attachment site at the 3' end of the molecule. It's the transfer RNAs that deliver the amino acids to the ribosome and read the messenger RNA to ensure that the amino acids are put together in the order encoded by the messenger RNA sequence. To begin translation, the ribosome must first bind to the messenger RNA so the sequence can be read and the protein generated. The binding of the ribosome to the messenger RNA is different between prokaryotes and eukaryotes, and it's the binding mechanism that allows the presence of operons in bacteria, but not in eukaryotes. In bacteria, there's a specific sequence of nucleotides that is part of the messenger RNA. This sequence is called the ribosome binding site, or the Shine-Delgarno sequence, and it lines up with one of the sequences of the ribosomal RNA in the small ribosomal subunit in order to bind. The small ribosomal subunit then scans the messenger RNA to find the first AUG codon, which will be the start codon. You can see the initiator transfer RNA here interacting with that start codon with its UAC anticodon region. This is different than in eukaryotes. In eukaryotes, the messenger RNA must be processed. A 5' cap and a poly A tail are added in order to get the messenger RNA out of the nucleus, and then the ribosome binds to the 5' cap instead of any particular messenger RNA sequence. This reason is the importance of operons in that the Shine-Delgarno sequence can be anywhere in the messenger RNA, and it can be in multiple places. So when we look at the sequence of an operon, remember an operon in the DNA has a promoter region and then multiple genes, so gene A, gene B, and gene C. That promoter indicates the start of transcription, and so when these are transcribed, we get one big long piece of messenger RNA with gene A, gene B, and then gene C. In front of each one of these, as part of the messenger RNA sequence, is going to be one of those Shine-Delgarno sequences. I'll label that SD for Shine-Delgarno. Remember, that's the ribosome binding site. So when we look at this messenger RNA to translate it, the small ribosomal subunit will bind to the Shine-Delgarno site. It will look for the AUG, and that's where it will start translation, and it will translate until we get to a stop codon, say a UGA. And in this case, we'll make a protein. But another ribosome at the same time could recognize that Shine-Delgarno sequence, find the AUG here, translate till we get to a stop codon, and make a different protein for protein B. The same thing would happen for the C region of the messenger RNA. This allows various ribosomes to interact in front of each one of these genes, get to the AUG, and translate to the UGA. If we took this same polycistronic messenger RNA, so a messenger RNA with multiple genes on it, and put this into a eukaryote instead, there would no longer be any Shine-Delgarno sequences. Instead, there's a 5' cap, and that is where the small ribosomal subunit binds to. That small ribosome scans until it finds the first AUG. It begins translation until it gets to a stop codon, so a UGA, and we would translate this first protein A. But when the ribosome gets to a stop codon, it falls apart. So the ribosome actually comes off of the messenger RNA. There might be a start codon down here for B, but this ribosome will never make it there because it will always hit the stop codon and fall off. Because there is no Shine-Delgarno sequence here, there's no way to start translation in the middle. We always have to start from the end at the 5' cap. And this is the reason you can have no operons in a eukaryote, because you would never be able to translate the remaining genes that are on that message. Once the small ribosomal subunit has bound to the Shine-Delgarno site and found the initial AUG start codon, then the large ribosomal subunit will bind to the molecule and form a sandwich around the messenger RNA. A new tRNA molecule will then enter the ribosome as what's called the A site for acceptor. This tRNA lines up with the codon at the base of the A site and brings in the next amino acid in the chain. The tRNA with the amino acid that was already in the ribosome is in what's called the P site for the peptidyl site, as this is where the growing protein is made. Once the new tRNA enters, the amino acid on the RNA of the T site is removed and it's transferred to the amino acid that's attached to the new tRNA. We can now see that connection here. The empty tRNA that no longer has an amino acid attached to it leaves the ribosome through the E site. The ribosome then shifts forward one codon on the messenger RNA, which opens up the A site over a new codon for a new tRNA to enter, and the whole process starts all over again. This will continue until a stop codon is reached. There are no tRNAs that match up with the stop codon, and thus the A site stays empty, which causes the ribosome to release from the messenger RNA, falling off and ending the translation of the new protein. Sometimes this is easier to see when the ribosome's actually moving, so there's another video on Canvas that's got an animation that might help make this process a little bit more clear if you're confused. Remember that we discussed mutations can occur in the DNA sequence either spontaneously or due to DNA damage by chemical or physical means. Those DNA mutations can cause variations in the genome between bacterial cells because one cell might have a slightly different spelling than another cell. But the DNA mutations only affect the organism's phenotype or traits it contains if the mutations affect the protein shape and function. There are different types of protein mutations that can occur due to the DNA mutations. DNA point mutations can lead to silent mutations in the protein. This is when the change in the DNA sequence, so we can see the point mutation here where this A has been changed to a thymine, but when you look at the codon, GUA encodes for valine, but GUU also encodes for valine. Since these are the same amino acid, the protein is actually not affected. So while there was a point mutation in the DNA, that leads to a silent mutation in the protein, and there's no change in phenotype because we have the same order of amino acids. Point mutations can also lead to missense mutations. This is where the change in the DNA, so here's our point mutation, causes the codon to change so that we have a different amino acid in the protein itself. So a missense mutation will change one amino acid to a different amino acid in the protein. These missense mutations can sometimes greatly affect protein structure. For example, if that is found in the active site of an enzyme, it's an important amino acid. But sometimes they don't have much of an effect. It really depends on the amino acid and its role in the protein shape. It also depends on how the amino acid has changed. So for example, if we changed a positively charged amino acid to another positively charged amino acid, it might not affect the phenotype much because they might have similar chemical properties. But if we changed a positively charged amino acid to a negatively charged amino acid, those probably will greatly affect protein structure and thus affect its function. The last type of protein mutation that can happen because of a point mutation in DNA are called nonsense mutations. This is when the point mutation in the DNA causes a stop codon to form where there hadn't previously been one. So now we form a stop codon, which means the protein ends here and translation doesn't continue. These typically will destroy the function of the protein because you only make, say, half the protein. You don't actually complete translation, which is why they're called nonsense mutations, because they typically will make a nonsensical protein that has no function. Remember, we also talked about insertion and deletion mutations in the DNA? Those types of mutations lead to what's called a frame shift mutation in the protein. Insertions or deletions will often change your reading frame as long as they're not in groups of 3. So you can see here, this TA that was originally has now been deleted. What that causes is we originally had GUA as your codon. We now have GCC because we've lost those 2 codons. Except all of the remaining codons are also different because now we're reading a different group of 3 since we lost those nucleotides. This changes all of the amino acids downstream of the mutation and can lead to the development of brand new proteins that sometimes function and sometimes do not, depending on what those new amino acids are going to look like. We've discussed a few of the differences between prokaryotic and eukaryotic gene expression, including the slight differences in the ribosomes, with 70S ribosomes being in prokaryotic cells and 80S ribosomes being in eukaryotic cells, and some of the differences in translation initiation. But another key difference between the two is related to the fact that prokaryotes don't have a nucleus. In eukaryotes, the DNA is housed within the nucleus. This means that transcription has to happen in the nucleus because that's where the DNA is, but the ribosomes are actually out in the cytoplasm. So after messenger RNA has been transcribed, it actually has to get processed. It gets a 5' cap and a poly A tail, and those structures say this is messenger RNA, and it should leave the nucleus to go to the cytoplasm, where then it will be bound by ribosomes to be translated. In prokaryotes, the DNA is actually in the cytoplasm already. And so this means that the transcription and the translation are both happening in the cytoplasm. And we say they're coupled together because they happen almost simultaneously. So as transcription is going on, as soon as that ribosome binding site, the Shine-Delgarno sequence, is made, a ribosome can come and attach and start translating. Typically, translation will start in prokaryotes before transcription is even finished, so the messenger RNA is still being made as it's beginning to be translated. This makes it a much faster process in prokaryotes because it doesn't have to go through all the processing and waiting to move from one compartment to another, like what happens in eukaryotic cells.